There you go. So this session is being recorded. It'll be made available to everyone who attended, but everyone who registered. It'll take us a couple of weeks to upload it because we need to add subtitles. So it takes a little while to process all the subtitles for all the taster lectures we've had. But whilst everyone's logging in, I think we've got most of our audience now. So as I said, if you just joined us, very warm welcome. If you have any technical problems, put them in the chat. Please put your questions at any point throughout the sessions. Please put it in the Q&A box. So I might answer it in text as we go through the session, or we might bundle them up until the end. But please post your questions at any point, and we'll have a Q&A session at the end. Somebody asked whether the slides will be available. No, but we will be providing a recording, so you can go back and review that afterwards. But if you want to search through it, with the fact that we're uploading a text file of this means that you will be able to search through it, hopefully on YouTube. I'm just going to launch a poll to find out. I'm just very interested whether you would be more interested in studying a degree or postgraduate. It really helps us to understand the composition of our audience. So if you could pop your answer in there, it'd be really great. Thank you. I've disabled this for the presenters, so don't bother trying to fill this in, John and Brenda. Oh. I know. So at the moment, it looks roughly like about 40% prospective MSc, prospective postgraduate, 45, 50%, which means it's roughly evenly split. So that's very interesting. It's changed from session to session. So thanks. It's just a rough idea to find out what level our audience is at. And we're just curious if you have an offer to study, whether you have an offer with us or elsewhere in engineering. This is part of the Spring into STEM series at UCL Engineering, so we're really interested to know whether people have offers with us or with somebody else. So I can see that coming in at the moment.
Or whether you've made a dreadful mistake and applied elsewhere. Or
So thank you very much for filling that in. So that's very interesting. So most of the people attending don't actually currently have an offer with us. So we're thinking your perspective.
But maybe you will come. Okay.
So maybe, so I'm very pleased to be joined by Professor John Ward and Dr. Brenda Parker. So I'm going to hand over. I will be here in the background. I may be answering questions. As I said, any technical problems, pop them in the chat and I'll be happy to help. So over to you.
Okay. Let's see if I can get this to work. No. How's that? You saying the right thing?
Great, thank you.
Jolly good. Okay, so John and I are going to take you through some of the concepts related to synthetic biology and the circular economy, and to look at maybe our two sort of slightly different but also complementary approaches to this theme. So in part one, I am going to cover a bit about bioprocess design for a circular economy and some of the work that we have been doing on photosynthetic organisms for this purpose. So if we look at what the IChemE, which is our kind of accrediting body in biochemical engineering, define as sustainability. So they talk about the triple bottom line, so that all chemical or manufacturing processes should, if they are sustainable, address not only environmental responsibility, they should also look at economic returns so that they are viable businesses or viable processes, and integrate social development. So what does that mean? Environmental indicators that can look at the resources that we use or the material that we dispose of. Economic indicators are the profits, and also that relates to the employment opportunities that we create. And social indicators are related to not only the place of our industry in society, but also the well-being and the things like employee turnover of people working on site. So are they doing valuable work and is it safe? So these are the things that we're striving for, and you'll see this kind of permeate through the lecture today in terms of the philosophies that we're trying to embrace with process design. So I wanted to introduce you to a few so maybe overarching topics which you may have heard about or maybe even indeed read about in terms of process design and how they might relate to the kind of things that we do. And so one thing I often see in personal statements that say people say they're very interested in biomimicry, but what does that really mean? So this is a philosophy that was really pioneered by Janine Benyus, and others that look at this kind of interdisciplinary cooperation of biology and technology. So a biomimetic design doesn't always involve biology. So in the case of Velcro, that's probably the most famous biomimetic example. But some of the really important things for me, and in terms of my interest in designing processes and designing with synthetic biology, is that nature runs on sunlight. So to me, that means that I'm really interested in using photosynthetic organisms. Nature recycles everything. So what can we use that is a waste product from one process that we can embed into another and upgrade? And also that nature curbs excesses from within. So as a process designer, I'm interested in how we can use biological systems as a way of regulating the kind of process inputs and the types of additional materials that we might need to add, and embracing some principles of green chemistry, which we'll touch upon. So yeah, as I mentioned, there are 12 principles of green chemistry. And for those of you who are interested in any kind of biotechnological or chemical engineering process, these are really important concepts for you to have in mind whenever you're designing anything. So 12 principles were listed by Anastas and Warner in a publication in '98. And it really talks about the idea that it's better to prevent waste than to go around cleaning it up after yourself, which I think that's reasonably obvious to everyone. But what it really sets out is a framework in which we begin to think about how to design inherently safer processes. So something can't leak if it's not there in the first place. And if we can try and replace some of the more harmful solvents with more benign ones. And the beautiful thing about a lot of biological systems is that they run on water, and that's one of the most, I guess, one of the safest solvents that we have. And in terms of chemical synthesis, if we can reduce many of the kind of wasteful steps in chemical processes, such as derivatization, where you may have multiple steps, and you replace them with something that's more selective. So for instance, using enzymes to perform a certain chemical conversion. We also should think about designing for degradation. And some of the things we're going to talk about today later on are a result of not designing for degradation. So in the case of plastics, there is no really easy and obvious end of life. And this is why some of the things we're going to talk about today are important. And again, the idea of using inherently safer chemistry to prevent accidents. So can we work at benign temperatures? Can we work at low pressures so that we can avoid the possibility of accidents, but also reduce the cost of our processes because we don't have to build in all of these additional safety and protective steps. So just moving onwards to get towards the point of a circular economy, you see the kind of theory behind where maybe some of the circular economy processes have evolved. So a really interesting book, if you haven't read it and you're interested in sustainability, is Cradle to Cradle, where it really begins to challenge the idea that, you know, the production process is essentially linear, that we should have processes that are integrated, that materials have both a biological cycle and a technical cycle. And we take responsibility for the disposal of goods, but also there may be upgrading into more useful components. And then we arrive at things like the circular economy. So if you would like to read and learn more about this, you can have a look at the Ellen MacArthur Foundation, who have published a lot and worked with a lot of industries on developing the idea of a circular economy for materials and processes. And what we're principally interested in talking about today is the cycle on the left-hand side, where we're able to use biological systems or biological materials to reform and remanufacture from a single starting point. So we're trying to really avoid ending up in landfill. That's really what, but what we're also looking at is atom efficiency and molecular economy, so that we're constantly putting our molecules back into good use, especially when they're particularly valuable ones or ones that have been quite costly for us to get in the first place. So that's just a bit of theory. And what I wanted to talk to you about today is some of my work in this sense, because you can really apply sustainability to almost anything. You don't have to be working just on kind of bioenergy or biofuels to be thinking about sustainable consumption or production. And in our case, what we wanted to know is, can we make the vaccination process more sustainable? I bet you didn't see that one coming. So in the aquaculture industry, if you wish to vaccinate salmon from a disease called salmon alphavirus, you need to pick up all of the individual salmon one by one and inject them with a vaccine. Now, I don't know about you, but that doesn't strike me as a fantastically satisfying kind of way to do things. So it's very labor intensive. It's also quite stressful for the animals. But it also creates an environmental footprint around maintaining a cold chain for the vaccine, and it's only suitable for some fish species. So the question we were asking is, can we apply a bit of disruptive design thinking to this, and can we look at oral routes of delivering a vaccine? So I'm sure many of you may not have had your vaccine yet. I've had one. But wouldn't it be wonderful if we could have an oral vaccination route? Because we don't need a certain amount of professional labor, less stressful, especially if you hate needles, and hopefully should be more environmentally and economically sustainable because you don't need that cold chain. However, what we don't know yet is exactly how the process works, whether or not we can obtain the same immune response from the gut as we can from injecting into the bloodstream. And it also is a bit harder to control the dosage. So in a fish farm, you don't know who eats what. So someone could be getting a bit of a double dose where someone might not be getting anything at all. To examine this, we looked at a model production system. So many of you might be familiar with synthetic biology in bacterial systems, but in this particular project, we are using a green eukaryotic microalgae called Chlamydomonas reinhardtii. And I guarantee probably within a few meters vicinity of yourselves, there's probably some of this swimming around in the soil or in the water nearby, an incredibly common freshwater species. So why do we use this? It's generally regarded as safe. So unlike some organisms that are known to produce toxins, we know that this is safe for consumption, which is important for a food chain. It's a model organism, so you can read an awful lot about the genetic tools that have been developed to manipulate Chlamydomonas, but also we've understood a lot about its metabolism and physiology. And we're able to do something called Chloroplast Transformation. Now, not something we can get into for a long time today, but essentially in the evolution of photosynthetic organisms, once upon a time, an organism has engulfed a cyanobacteria. And that cyanobacteria really became what ultimately known as the plastid or the chloroplast. But it has a small amount of DNA inside, and it's exactly this that we're able to use for our synthetic biology work. And because the genes for photosynthesis are expressed what's known as constitutively, so they're switched on all the time, it means that any proteins that we want to express are also being made all the time too. And because we're expressing genes in the chloroplast, we have this natural bioencapsulation, which hopefully will enable our vaccine to be delivered safely to the gut and not digested by the fish in the process of getting there, or meanwhile. So our project looked at a number of different things, and I guess any design project in this space will probably follow a similar pattern. So what we want to know as biochemical engineers, what is the relationship between the sort of synthetic biology, the construct design, bioprocessing, and then ultimately the vaccine stability and performance? And how can we design a sustainable strategy to manufacture this vaccine as well? So we undertook genetic engineering of Chlamydomonas to be able to produce a number of vaccine candidates. We have undertaken some fish trials at a place called CFAS on the south coast. So they were able to test the vaccine construct that we made, and then look at some of the pathology results to see if we're getting the immune response that we want from the challenge trials. Meanwhile, back in London, we're doing a bit of process optimization. So we're looking at some of the challenges of scaling up, which I'll tell you about in a second, and also thinking about our techno-economic analysis. So again, revisiting this idea of the triple bottom line and how much it might cost to produce such a vaccine. So for those of you who might be interested, these are some of the constructs that we made. So we know to be able to induce an immune response, we need to have a certain section of the virus called protein present. We're looking at different regions of this. So what is a big challenge for us is how big of a molecule can we express in the chloroplast. So there are certain limitations. I think the SAV4 is one of the biggest proteins that have been expressed in the chloroplast to date. And as a result, the expression was rather low. So what we want to know is, are we able to see the immune response with only a portion of this protein sequence? So we were using a process developed by Saul Person at UCL as well. I don't have time to go too much into detail about this, but we were able to insert the gene coding for the SAV proteins into the chloroplast using a really elegant expression system where we would restore photosynthetic functions. So we were able to select really precisely for only the transformants that had the gene of interest. And after that, we began our work on scaling up. So here we're growing algae in our kind of 5-liter hanging bag cultures. You can see after 3 days, it's reasonably nice and green. Unfortunately, even though algae look very green, there's not very much there. So there's about 0.5 gram per liter of dry cell weight. So because it's so heavily pigmented, it's quite misleading. And then we were looking at whether the cells were healthy as they were being grown. So one of the challenges as a bioprocess engineer that we are facing is, yes, we can engineer cells to produce all kinds of interesting proteins, but we also have to look after the cells and make sure that they're healthy and not getting broken, and what's point in the cell cycle, at what age the cells have to be whenever we harvest them to be able to express our protein properly, but so the cells are still intact and not beginning to kind of fall apart. So then we ask ourselves, okay, we've grown our cells, what are we going to do next? So we know that our vaccine is inside the cell. So what we need to be able to do first is remove the water. So as I showed you just in the slide before, we scale up our culture. Then we look at ways of harvesting. And we have to maybe ask a number of different questions about what we're going to do. So we have a lot of water. And even though algae are amazing photosynthetic organisms at taking CO2, it's going to take a lot of energy to remove that water. So we have 3 options. One is to centrifuge the cells, so to use gravity and to separate them out in a centrifugal device. Now, this can be a bit problematic because our alga has a very fragile cell wall. So we may damage our cells. So we have to do some studies to understand the conditions in which they will tolerate. We could look at filtration, which is a more gentle way of harvesting cells, but it has a limited concentration factor. So we may have to do more than one filtration step to actually remove the water from the biomass. Or we could look at flocculation with chitosan. And now, this is actually a very elegant way of doing things. Chitosan is a byproduct of things like shrimp processing, so this is a natural product. However, this sort of breaks one of our rules of the 12 principles of green chemistry, which is to try not to add anything additionally to a process. So this is something you're going to have to remove later, and that creates a footprint of its own. So we have some ideas of how we're going to do this, but it's certainly not going to be easy because, as with everything in bioprocessing, there's always a little bit of a trade-off between the simplicity of the process and how well it works. And so this was what makes for really enjoyable research, I have to say. And then when we have the wet paste of algae, we need to work out how we're going to dry it, because what we want is a kind of freeze-dried formula, a bit like freeze-dried coffee, if you've ever seen that. And so we have 2 ways we can do this. One is spray drying or freeze drying. Spray drying is much more compatible with large volumes. It's a cheaper process because the equipment is not so complex, but we're able to achieve a higher throughput. So what our research wanted to ask from this is, if we use spray drying, will we damage our vaccine product? So we ran our process with the hope of answering that. So this is a Western blot here. You can see the band of the kind of product size of interest. We can see that our product is found in the cultures in what we call a slurry, the harvested biomass from the centrifuge. We see it in the spray dried powder and in the freeze dried powder. So essentially we see not a huge significant difference between the spray drying and the freeze drying. which is fantastic news for us because that really simplifies our process. And now, thanks to lockdown, we've been able to do our stability studies because we couldn't go to the lab. So we prepared the powder and we've left it on the bench. So we'll be able to see if our vaccine is still active after all this time being stored at room temperature. So just to maybe change gears before I switch, hand over to John, is in my other role, I am a director for a program called Biointegrated Design. And we use algae in a number of different ways, actually, to look at circular economy. And in this particular project that I'm showing you, it's called INDES. So this is led by a PhD student called Shanil Malik. And here we were using microalgae as a means of capturing heavy metals, so making use of some interesting molecules that they produce called phytochelatins, which are capable of binding heavy metals. And here we were trying to re-question really the idea of what the bioreactor might look like. And is it always going to be a kind of stirred tank vessel, or can we embed some elements of design so that we could work in maybe places where space is constrained? So for instance, in these kind of small scale artisan manufacturing sites. So this is a wall that that she had built from these tiles that have algae embedded in a hydrogel inside. And another project which we have been working on is to make use of the fact that many algae produce something called EPS. So this is exopolysaccharide, and this is a sticky substance which often enables them to adhere to a rock or a benthic surface or whatever. But in this case, we were growing them on a textile and looking at how we can capture microplastic waste. And this gives us maybe some other freedoms to design with the amazing properties of biology, but to look at overcoming some of the limitations associated with using conventional processing equipment. So this is some work from Annette and Jin and some colleagues looking at attached cultivation of microalgae. So again, using light and growing in ecosystems with very simple nutrients, but being able to capture microplastic waste thanks to the kind of bluey substance that they produce. But once we capture the microplastics, what are we going to do with them? And I think John is going to tell us what that might be. So John, I'll hand over to you.
Thank you very much. Hi, welcome everyone. I'm John Ward. I'm a molecular microbiologist and have lots of interests in using enzymes to both make substances, but also to chop things up and to recycle them. And that's what I'm going to talk about this afternoon. Now, Brenda has control of all the slides, so yeah. I'm going to have to keep saying,
there we go.
Oh, next slide, please.
Okay.
That's fine, lovely. So I'm going to talk about recycling plastics, and specifically the example will be PET, polyethylene terephthalate, in this part of the lecture. And plastics, as everybody knows, are really advantageous materials. They've only been around for probably 60, 70 years, and they have replaced lots of the things that humans used to use from several thousand years before that. Ooh, yeah.
Okay.
And what I'm going to talk about in the next few slides are PET, and everybody's familiar with PET bottles, polyethylene terephthalate, the water bottles that everyone seems to carry around, but they're also used in a whole variety of other food containers and many other materials as well. In fact, quite a lot of them find their way into textiles, but we don't often know that they're there. And those things have replaced glass mainly. And polyethylene bags replaced cloth and other natural materials, and they've actually now been in the UK and in Europe phased out of being just given away at supermarkets because of our worries about them ending up in the environment. And then there's other materials that you'll also be familiar with. Nylon's been around for quite a long time. It's in many, many textiles, but it's also, when it's formed into hard material, it can be used in car engine parts and the moldings in cars and things like that. It's very tough. And then other polymers like lycra, which is stretchy, polyesters, when you have mixes of those and all the different threads, they've replaced cotton and wool for clothing, and they give clothes that sort of stretch and the sort of grip that people like. And many other plastics. I won't go into them all, but our lives and our surroundings and all the things that we use are largely composed of plastics now. There's a bit of glass and bits of metal. But all these plastics were developed years before we really thought about what would happen at the end of life. And in fact, the end of life for a lot of products is now being designed, as Brenda talked about, into the whole idea of the product, rather than let's make lots of this stuff and okay, well, we'll not even think about where it's going to go. And of course, all these plastics ended up in waste, in landfill, and that leached out into the oceans and the rivers. Hence, in the last couple of years, people have been really worried about this continuing, and lots of curbs have been put on that. So Brenda, can you take me to the next slide? So the advantages of plastics and PET is it replaces glass, and that actually is quite a major advantage. The energy costs in transporting glass are really quite a lot. And if you then think about the use of fossil fuels in all the kinds of transport, PET is actually a very valuable material, but we shouldn't just throw it away. We should be trying to recycle it. It's quite resistant to degradation. So when it gets into an ocean or landfill, it's there for a long, long time. And that gets abraded, and then you end up with microparticles. And those are the things that then are giving people a little bit of a worry, because they end up even more dispersed and actually in the food chain. So polymers are great, but we really need to now consider how they're going to be recycled and their end of life. And in fact, they'll have multiple lives for some of these plastics. The disadvantages are written up there. I'm not going to go through all of these things because I've been mentioning about they just actually end up in landfill and in the oceans. Also, we must remember plastics are actually based on oil. And in some parts of the world where a lot of stuff's been dumped, they just burn it. And that leads to a net CO2 increase in the atmosphere. If we go to the next slide, this is a survey or a study that was done by the Earthwatch Institute a few years back. And they looked at European freshwater environments and just collated all the things they could find and quantitated them in sort of percentage of objects. And you can see there that plastic bottles are 14% of all the things they found. Now, if you look at some of the other things there, some of the food containers, wrappers, cups, they will also contain polyesters. Some will even be polyethylene phthalates, plastic straws as well. So the PET going into the environment is really quite a major amount of these waste plastics that are going to take ages to degrade if they're not, if they're just put into the environment. So if we look at the next slide, this is something that shows what me and my group, we often go to odd places to try and get new bugs, new bacteria, and maybe bacteria that are unusual or have been exposed to some of the materials that we're interested in and maybe have even started to grow on them or degrade them. So I learned about this from a TV documentary a few years back, where they were looking at old waste sites. And this was an example. And it's down by the Thames. It's beyond London. It's more to the sort of seaward side of London. And it's where in the 1950s and the 60s, it was considered sort of miles from London, big open landfill. We could fill, it was probably a gravel pit where they'd taken out a lot of gravel, big hole in the ground, and they decided to fill it up with rubbish from London. And that's happened all around the major cities in the UK. And these were then buried with some soil cover, and then grass grew on top of them. And in fact, parts of this, I'm not going to show the pictures, are actually farmed. And you can see just a bit over from the banks, there's some of this is underneath farmland now. But as the years have gone by, and this was last actually used in the 1960s, then it was capped, covered with soil. But the edges of the Thames have sort of encroached with storms and rising water levels. And now this is exposed. This part here, if you were looking at this, to your left would be the edges of the Thames. And we went down at low tide, otherwise we'd have got our feet wet. And it's basically, it's the shoreline of the tidal part of the Thames, which at high tide is actually touching the bottom of this little bit of grass here. And on stormy times, it's really degrading the edges of here. And what you can see are layers of all the compacted material that, what would this be now, 50 years ago were put in there. And you can drag out bits of whole plastic. Bags that were used in those days. There's not so much of the modern day plastics, but there are all kinds of recognizable plastics, materials, but also metal. There's a lot of weird containers there that we're a bit wary of, because in those days anything was dumped into landfill. Now this just shows that even in a landfill site where people think, oh, well, this will degrade in the soil, 50 years later, you can pull out, in fact, we pulled out some newspaper that you could read typeface on. So things just do not degrade when it's compacted and there's no oxygen. So there's not a lot of the PET waste there, but just imagine what will happen in 50 years from now. Where's all the PET bottles that we're going to be putting into landfill? Well, a lot of them are mechanically recycled now, but we'll have a lot of landfill where things like this will be obvious in 50 years' time. If we go to the next slide, this is just a couple of more pictures of really discrete bands of all this material that were put in and more soil. And you can actually trace through lots of different loadings of this landfill site. And you can see then on the bottom picture, the actual look of the more sort of stark part of the Thames where it gets flat and goes out to the very wide estuary. And there's Maria, one of my postdocs, sampling, and we went there with about 4 people and we all took samples and they're still stored. And we've been looking at the bacteria contained in some of those samples. It takes quite a while to go through all of those. So can we go to the next slide? So I'm going to talk a little bit about what PET actually is and how we might start to recycle it with enzymes. So PET is short for polyethylene terephthalate, and it is, as I mentioned, it's become widely used in many different applications, and we know about it very obviously from PET bottles, but it's used in many different types of materials in the textile industry as well. Now, PET bottles and food containers can be 100% recyclable by what we call mechanical recycling. If we go to the next slide, this is what I mean by that. Now, PET, because it's easy to recognize, the objects are quite big and they're very separatable by us in our kitchens and by these facilities that are called MRFs. Now, I can't remember what it stands for. Anyway, these are huge facilities where all the rubbish goes, all our recycling goes, and through a series of conveyor belts and infrared scanning to actually, at very high speed, detect PET, and then it's blown off into another conveyor belt. I went to one of these a couple of years ago, and they're amazing facilities. Multiple conveyor belts all moving around. Eventually, what happens at the end is you've got a purified big pile of PET bottles and other PET, and other different elements of the rubbish have been separated and concentrated, basically. And then it can get compacted, the PET, and it goes off to a factory where it can actually get melted down again and recycled. So that's called mechanical recycling, because PET, certainly the bottles, are easy to spot, to recognize, and to separate in one of these facilities. So at the moment, in places where a lot of recycling is actually collected, so many London boroughs, many other places around the UK, that happens. And quite a lot, a high percentage, not every piece of PET that goes into your recycling actually comes out into a big concentrated bale that can go to a factory that then will actually make new PET bottles. But there's always a loss in that system. So the cycle isn't a closed loop, what we call a closed loop, where 100% of the material goes round and round all the time. There's always some loss. And also when you mechanically recycle PET, you actually degrade or break the polymers a little bit each time. So at every time you use a bit of recycled PET bottle, you have to add what we call virgin plastic into there, really good high quality plastic, so that you keep the optical quality and the mechanical properties of the bottle. But PET is eminently recyclable. If we go to the next slide, even though it is eminently recyclable, it's not 100% recycled everywhere around the world. And as you know, lots is in the environment. And so researchers in several parts of the world, and this was the first evidence of bacteria that could degrade or grow on PET, have been looking for bacteria or enzymes that could actually use PET as a carbon source. Now, bacteria are hugely versatile, bacteria and fungi, hugely versatile, and they'll look at anything really that's going into the environment as a potential food source. And very quickly, because they reproduce quite quickly, you will find bacteria that will be able to at least grow on a little bit of the material you put in, unless it is really nasty and toxic. And then in 2016, a group in Japan showed that they had isolated a bacterium that could actually grow on and very slowly start to degrade PET. And you get from the degradation of the PET phthalic acid and the ethylene glycol, which is the actual monomers used to make PET. The bugs will then go on and they've got pathways that will actually eat up those compounds, but it's easy to trace the steps in the pathway. And then other people isolated fungi. So there are quite a lot of bacteria and fungi in the world that can actually grow on PET, even though it's only been in the environment for probably 50 to 60 years, and in any large amounts, probably only the last 30 to 40 years. And it shows you how quickly bugs will develop and evolve to be able to grow on those things. But that's not quite a process to recycle things, because these grow very slowly. If you go to the next slide, Brenda, this is what we would like to do. Take some very specific enzymes from these bacteria. They could be from there, could be from other sources that are specific for the bonds that join all the components in PET, the phthalic acid and the ethylene glycol. And if we can chop those, we can then release those monomers again, and we could recycle them, or that we could use those as a source for other compounds, other bits of chemistry. So it could be considered then as a resource. But you need some really good, powerful and specific enzymes. And that's what we've been trying to develop in our labs. We're not the only ones. Many companies and research labs are doing this because the prize is very valuable. I mean, there are companies now that are offering to recycle all of Coca-Cola's PET bottles in Europe. And that's probably starting to take place. A bit overhyped in some of these things, but thank you. That's so PET is an interesting molecule. It's a polyester. Now, a lot of natural molecules are esters. Fats are esters. And so there's a lot of enzymes in the environment that could conceivably take this ester bond and then hydrolyze it, add water across that ester bond and open it out. But it's the molecules on either side of that, these big aromatic molecules, that are actually a bit different from some normal esters that might be around in the environment. And if you could chop up PET with the PETase, the enzyme that came from this bacteria, you would get the variety of the monomer compounds. TPA and ethylene glycol, the ones on the right-hand side, are the real monomers. But if you chop the ester bonds in different places or the polymer chain at different places, you can get either MHET or BHET with either 1 or 2 ethylene glycols on either side. But more of the PETase would then degrade it even more. Can we go to the next slide? Well, that's just showing the ester bond. The PETases have had quite a lot of work done on them, and even to the point where we know the 3-dimensional structure of this enzyme, which is quite a sort of complex thing to develop, quite an academic tour de force to find this out. And it turns out that they are similar to already known enzymes called cutinases and some lipases, which exist in the environment anyway. Cutin is the very hydrophobic material on the surface of plant leaves, which makes them hydrophobic. And if you watch water, raindrops falling on a nice glossy plant leaf, the water will bead up into a little bead. And that's because there's a thin layer of this material called cutin on the surface. So the cutinases that are made by bacteria that break down plants in the soil are similar to the PETase. And in fact, the PETase has probably evolved from a cutinase. So if we go to the next slide. So what we're trying to do in a grouping at UCL called the Plastic Waste Innovation Hub was to develop enzymes for recycling plastics. And we started off with the example of PET, and we have developed ways of looking for new enzymes. So that's metagenomics and genomics, where we've used the protein sequence of an enzyme, in this case the PETase, and put it into databases electronically. You can find tens of thousands of genomes and databases on, they're completely public, anyone can do this. And you can look for homologies, and from that we can then fish out and then make or clone new different types of PETases. And metagenomics is just a sort of a bit more complex way of doing that with mixed genomes, but I won't go into that. We're also interested in thermostable enzymes, because if you could do this process at a high temperature, there's quite a lot of advantages in using enzymes that are faster at high temperature. It stops other bugs trying to develop and grow on these materials. If we can go to the next slide, and we'll whiz through some of these. So this is just a schematic to say how we make better or look for better enzymes. So we have a sort of a cycle. It's just shown as a line here. We have the parent gene. So this would be the first example, but we have many other wild type PETase genes. And what we do is we copy that using enzymes that actually put mistakes in. And we do it so that the mistakes are only 1 or 2 per long gene. And that's effectively making a mutant. So that's our mutant gene library. Now, many of those will be inactive, but we don't mind, because we're dealing with numbers that are in the sort of order of 10 to the 9 different DNA molecules. They're put into a bacterium so we can see their activity, and we do what we call screening of those. So we have sort of a bit of automation to look at zones around colonies that might be degrading some PET that we put into an agar plate. So that's one example of how we look for better enzymes. But we have to go through this cycle several times, and we take the best ones and we look at those, assess whether they're any good, any better than the original one, and maybe use those as a next stage of making some mutants. And eventually we have some evolved PETases so that we can test against native filaments or film or fragments. And that's still going on. And in fact, Maria Bourne, who I mentioned or showed a picture of in one of the early slides, is still involved in that project. And we're getting ready to sort of write this up and publish papers on many new and highly active PETases. And what we found is that these PETases are actually quite good esterases for other kinds of plastics, which are themselves different esters. So we're looking at lycra, which is a coester, it's a polyurethane and an ester. So by developing enzymes that are really potent and robust on one material, they may have good properties on other similar materials, and that's what we're looking at the moment. If we go to the next slide, this is another way that we were using to find new enzymes, not only the petases, but we've got other hot bin composters that we're looking at nylon and lycra degrading. And this is where we simply took PET bottles, food containers, we cut them up, shredded them, and then we put them into this compost bin, which is called a hot bin. And it's got thick walls of polystyrene, another polymer, but that's very insulating. And so the internal chamber is not that big compared to big composter heaps, but it will retain all the microbial heat that bugs put out, and we put out heat as well. Microbes do, but that is usually just dispersed straight away into the environment. But if you have quite a lot of volume with bugs growing on food source and you completely insulate that, the temperature will go up, and it goes up to, can go up to 60 or beyond. And the bugs that you get there are very robust, and we'd like to use thermophiles for some of the processes. I think somebody asked a question about, won't having high temperatures be a disadvantage? Well, we are looking at different temperatures as well, but thermophiles are quite useful for doing quite a lot of operations. So from a hot bin like that, we can isolate bacteria. We go back to our labs. This is only about 100 yards, 100 meters away from our lab, sitting in Gordon Square, just near UCL. And from this, we can get bacteria. We can then isolate genes from those, metagenomes, and hopefully get new petases. Can we go to the next slide? I won't go through this in any detail. This is just showing how we clone genes. We've got the DNA, we'll chop it up, we'll put it into little plasmid molecules in our test tube and then introduce those back into bacteria. That's basically how we go from genomes that we've isolated, we've extracted from the bacteria growing in a hot bin or a soil or that landfill, and eventually we get them into an individual E. coli cell in the lab, and we can then study that. Can we go to the next slide? Okay, can we go again, because that's just showing the process of transformation. And this is revisiting that bit of the slide that I showed earlier, mutant gene libraries. We have lots of picking of colonies from the initial screening. And then we often now use activity screens with esters that sort of mimic PET in one half, but have a color compound on the other half that's only colored when it's been released by a petase. And then this color comes up and you can see some sort of vaguely reddish colors in the middle plate there. And we use these multi-well plates quite often now, and the automation can start to add different colonies to each well. We add the reagents, incubate it, and then after a few hours we see, oh, these are the really red ones. We can then pick those and then study those, look at the gene sequence at that point, and we see what the changes are. And part B on this slide shows the actual output of one of those DNA sequence runs that we send the genes off to a company, they'll sequence those for a few pounds and send us back data like that. And from that we can start to see, oh right, in our normal PET we've got these mutations here, they're near the active site. There's ones here that are with outside the active site, and they may be acting on sort of a flexible bit of the molecule to make it less flexible. So we start to learn a lot about this complex protein. And once we've learned enough, we can start to deliberately put in new mutations that we didn't isolate, but we can say, okay, this is a point that was isolated as an alanine change in the mutant. What happens if we make lots of different changes at that point? And we can sometimes discover even better enzymes that way. And then if we want to go to make a lot of this, to actually make a real process to develop an enzyme that could be used in industry, we would have to scale up. And this is what UCL Biochemical Engineering is preeminent at. And the degrees and master's programs at UCL actually have a lot of this the steps here to go up to the large scale production in our fermentation facility. And we have many fermenters and all the downstream processing equipment to take bacteria and their enzymes through to mimicking a process that would happen in industry, which is, and this kind of thing is very unusual and quite unique to UCL. So we go from that small scale that you've seen in those screening plates, little flasks, bigger flasks, and eventually into big fermenters. And in industry, they will have even bigger fermenters to produce multi-thousand liters of enzymes. I wonder if that's the last, oh yeah, so going to a circular economy, we could have even slightly different route for this. So growing agricultural material, food, often leaves a lot of waste behind, which goes into the soil or gets composted, but some of that could be used. It contains cellulose. We're not using the food part. We're using stems, stalks, and bits like that. The cellulose can be fermented. So it could go through a process of chemical and biochemical engineering to make biodegradable polymers. And we are doing that in our group, but PET could be considered biodegradable. At this point, it could have an arrow coming in if we have the enzymes that could degrade it. And then you could go through a cycle of having packaging that is collected, it's identified, and it's composted or degraded in a big factory. And the material could then either go to being used in making more plastic, or it could go back into the soil. So this is part of another kind of cycle where we consider almost a sort of fully sustainable loop from growing material using photosynthetic energy and CO2 all the way through to our materials, our plastics, and at end of life, it gets composted, maybe with added enzymes and bacteria, and it goes back into the soil, or key bits of the material can be used again. Now, I wonder if that's the last slide, Brenda.
It is indeed. It is indeed. So, yeah, I guess now we have time for just a few questions. And
just before, sorry, just before you go with that, I just want to remind people that Brenda will be at the UCL Undergraduate Open Day on Monday. So I'm about to post a link to that. If anybody would like to find more about the degrees, please sign up to it. We've got sessions at 12 o'clock and at 5 o'clock. So just a quick little advert from me.
Yes,
and that's
I think we have some high schoolers here. So yeah, definitely. If you want to come and join the undergraduate, I'll happily see you again on Monday for a different talk. But all right. One interesting question that's here, John, is about estimation how long things take. And I think that's a really good point in our industry. So obviously the plastic in the landfill has been there for many, many years. Someone was wondering how long this might take compared to the generation of new plastic products. Is it something you guys are working on at the minute?
So how long does which part take?
The degradation of the plastic.
Well, it does depend on where it is and the environment. I mentioned this particular landfill, it's been very compressed. There's no air getting to it. Mm-hmm. So it's partially anaerobic. There's probably not a lot of water getting in. So it's not a very good environment for rapid degradation. If you took the same material out and separated it, you could blow air through it and mix it up. Parts of it, the paper that we could find in there that was still quite readable, would degrade very quickly because you're actually giving it all the right conditions. So landfill and compressing it is often not the best way to deal with many of these kinds of slowly degrading material, unless you deliberately want it to slowly degrade. I mean, from landfill, we get methane, which can be tapped off, and you can make energy directly from that. But the timescales for some of the plastics, if we had enzymes or bacteria that could grow on those, and this is why I was thinking and talking about thermophiles, they grow and degrade things very quickly. There's very active degradation. Things are more soluble at higher temperatures as well. So if we could take some of or separate some of the stuff that had gone into that landfill and then put it into the right conditions with the right bugs, then you would get degradation much faster. So the degradation just depends on the environment, how you're then allowing the bugs to attack and to grow on the materials. Somebody asked about, wouldn't creating higher temperature for enzymes just lead to more fossil fuel use and air pollution? Well, yes, if you then burnt coal to make your things grow at a higher temperature. But when I was talking about the hot box, when you have a lot of bugs growing in a confined space, the main thing you have to do is to cool them. What I didn't show or point out in that 450 litre fermenter was, you do have heating coils in for certain things, but you have a lot of cooling coils. Once the bugs start to really get growing, their metabolic heat is trapped and it can't escape very quickly from the surface area of the fermenters. You have a lot of cooling coils in there. So it's actually quite easy to get bugs in a big fermenter to get hot. You don't have to put in much energy. The energy goes in the spard, keeping it mixed, and often a little bit of cooling as well. But modern day fermenters are able to cope with thermophiles, and so you could grow things with very little input of energy at 50, 60 degrees.
Yeah, I think we also have to bear in mind when we talk about high temperatures, when we're talking about biology, I mean, we're usually talking about less than 100. I think Absolutely. If you go to any other branch of engineering, high temperature is somewhere, maybe 500-600 degrees. So we're really, if we think about it in the context of things, we're not really working. It's for us, it's high temperature if something goes to 50 or 60. But yeah, in the context of wider engineering, it probably is still quite mild conditions, isn't it?
Yeah.
I think that's the first question, which I think if we have time, maybe I'll answer quickly and then I'll hand over to John. So there's a really interesting question about how did you come up with the idea? And I think this is a lovely question because I never get asked that kind of thing. So thank you very much to the person who asked it. So for the edible vaccines project, we wanted to understand synthetic biology in photosynthetic organisms. Now they grow a lot slower than compared to John's bacteria. And there's a lot of the things that are not quite as well known compared to cloning and expression in bacterial systems. So we wanted an application where we wouldn't need a huge quantity of our product. And there had been some previous work with a group in Thailand looking at the aquaculture industry there. And we were looking at, how we could perhaps embed this in a bigger conversation about sustainable aquaculture. So as usual, it's often a happy collision of circumstances where you may be working on something, someone has a conversation with you, you realize that's a good application. And then there was a sort of funding call from a funding body called NERC, who are looking for new innovations in aquaculture. So that was an opportunity for us to pitch this project and get it put forward. And it just so happened that we had been working the year before on making a kind of antimicrobial called endolysins in microalgae. So this seemed like a great vehicle to be able to test some of the things that we had learned in a different application. So yeah, but John, how did you get started working on plastics? What made you decide to do that?
Well, I suppose it was just growing out of the ideas that we'd been looking at an interest in natural polymers, bacteria that could degrade, first of all, things like protein, a lot of very interesting proteases. And we look at those on these kind of agar plates where if you've got a colony growing, you can see a zone around it if it's able to secrete an enzyme that then degrades a material you've put into the agar plate. And that could be milk protein. We see these zones of clearing. And we got into, over the course of many different projects, agricultural waste. So there was the use, we were looking at waste sugar beet. When sugar beet is grown in the UK, you extract the sugar, it's sort of like a hot water extraction, and you end up with all these little, what they call chips. Well, they're dark grey, but they're chip shaped. And they are cellulose, they're a lot of pectin, and they're just sent off for animal feed. But in fact, in the UK, there's not enough animals to eat those. So they were just being, well, they were such a cheap waste product, but they'll go back into the soil. But there's a lot of valuable polymer and carbon in there. So from that, there was a whole project with several universities developed to look at how we could use different types of enzymes to degrade those. And those are natural polymers. Remember, cellulose, plants grow and are big plants and trees by making very strong structural polymers and biomaterials that have all kinds of linkages between the pectin, the lignin, cellulose. So this kind of idea of looking for enzymes from bacteria to degrade really recalcitrant or slow degrading polymers has been, in my group for many years. We looked at other kinds of materials as well over the years. And then when people started to be really quite concerned about plastics, especially microplastics, which we're still trying to get a handle on, and Brenda's got this great idea of trying to trap those before you then do something with them. So I started to develop the hot bins actually for the PET and nylon and lycra before we got funding for the project. And then we applied for funding for that, and we got that. So it's got lots of tendrils looking out for being able to degrade these quite recalcitrant materials in the soil.
Yeah. So I think the short answer to your really, really profound question is probably by observing the world. And I think this is the key with synthetic biology, that if you can observe something in nature, and coming back to the biomimicry principle, is can we then turn that into something that is a process? I think we have to end there. I can see Kim is watching his watch. We're after 2 o'clock. So yeah, maybe you want to wrap up, Kim.
Yeah, well, thank you very much to everyone who's joined us. Thank you for the questions. They're really great set of questions. And as ever, we're sorry we couldn't get through all of them. Just a reminder that Brenda will be around on Monday. I've also sent a link to a questionnaire. So if you've got a few moments afterwards, if you could click that link and fill in the survey, that'd be really helpful to us. But thank you very much. Thank you, John. Thank you, Brenda. This is the final taster lecture in our series. So thank you everyone who's attended, and a recording will be made available afterwards. Hope you have a great afternoon. Thank you very much.
All right. Thanks for joining us.
Bye, everyone.
Bye.
Bye.